Welcome back, students. Welcome to Priya's Life Science Academy. This is your educator Priya. Today we will learn another very important topic of inheritance biology, genetics. This topic is very much important for those who are preparing for CSIR NET Life Science, ICMR, NEET, and also for the students studying in bachelor's degree and in master's degree. So let's start. So the topic is complementation test. The term given by Samuel Benzer. It is used to detect two mutants are in the same gene or not. Or if I say in a very simple term, I will say that it is used to detect whether a characteristic feature is determined by two genes or one gene. Okay, whether a feature is determined by one gene or two gene. Okay, so this is the main basic funda of complementation test, whether a specific characteristic is determined by more than one gene, or you can say it in a different way, that whether two mutants are in the same gene or not. So two different mutation or gene of interest occur in the same chromosome, gene, or not. Or we can say two different mutation or gene of interest occur in the same chromosome or gene, or not, that can be studied with the help of complementation test. So its name saying that it's complementary to each other. Okay, so if genes are complement with each other, that means the genes are present in different chromosome or gene. Okay, if they do not complement, that means the genes present on different chromosome. Okay, so if you see that two genes are complementary to each other, that means they are on the different chromosome. And if you see those two genes are not complementary to each other, you can see that they are on the same chromosome. When I'll give you the example, it will give you a clearer idea. So when the two mutations are on the same chromosome, the arrangement is called the coupling or cis configuration. I told you about the cis and trans when I taught you lac-operon, and a heterozygous with this genotype is called cis heterozygote. Okay, and when two mutations are on the different chromosome, it is called trans configuration, and an organism with this genotype is known as trans heterozygote. So this is the example where you can see that these two mutations are on the same chromosome. M1 and M2, these two mutations are on the same chromosome. So this is cis configuration. And here you can see this M1 and M2, these two mutations are on different chromosome. This is known as trans configuration. Okay, so whenever the mutations are on the same chromosome, this is known as cis, and whenever they are in the different chromosome, it is known as trans. They are also heterozygote because M1, M2 two different kind of mutations. So this is cis heterozygote and this is trans heterozygote. Okay, and point to be noted, very important point, this complementation test is only applicable for homozygous recessive condition. Okay, homozygous recessive condition, because only when then we can understand the effect of a mutation. Okay, if small a is there, we can understand that there is some kind of mutation, because for a recessive gene to be effective, it needs a double dose. So always remember that complementation test is only applicable for homozygous recessive condition. Now coming to the example, I am considering the feature of Drosophila's eye color. So I told you the best thing to understand complementation test is through by this example. Okay, so I told you in the first that you have to know that whether a characteristic feature is determined by two genes or one gene. So let's say the eye color of Drosophila. For example, this is the white color. When you have pigment A, that means when you have the action of gene A, pigment A means gene A. From gene A, pigment A comes, it gives white again, and when in this white gene B works and it gives pigment B, then the eye color of Drosophila transforms into red. So here two genes are working simultaneously to give the red color. Okay, so if there is red eye color of Drosophila, so we can say that both A and B pigment is present. So the eye color of Drosophila is determined by two genes, gene A and gene B. Two genes determine the eye color of Drosophila. If there is white eye color of Drosophila, so we can say that either A or B is absent. Okay, either A or B is absent, then you cannot get the red eye color. So we can say that A and B pigment are complementary to each other. So both gene A and B's dose are important for determining the eye color of Drosophila, to get the red eye color of Drosophila. So here you can see small a, capital B when crossed with small a, capital B. So this fly, what you can say, this fly is white in color. Its eye is white. This fly's eye is also white because there is small a and capital B. You need both capital A and capital B for the red pigment. So why these parents are white? Here these parents are small a, capital B. They are white because in order to produce the red eye color, you need both capital A and capital B. There is no capital A. In this case, there is small a and capital B. But if there is small a, and capital B can't give you red. In order to get a red pigment, you need here capital A. Again, for this parent also, they are also white in color. Why? Because even if there is double dose of capital A, the B's are small. So they are also white. Here the case is there is capital A, okay, but small b. So no red color. So in order to get a red eye color, you need both the gene capital A and capital B. So both genes, they are complementary to each other. That is why we are getting red pigment. So this is known as complementation, when a characteristic is determined by the combination and complementation of the two genes. These two genes together work and give us the red eye color of Drosophila. So when you cross this white parent with this white parent, what will you get? So white parent, you will get a red offspring. How? Because this is white parent, small a, capital B. This is again white parent, capital A, but small b. When gametes are produced, what type of gametes will it produce? Small a, capital B. Here gametes will be capital A, small b. So when offspring is produced, it will get a capital A from this parent, small a from this parent, and capital B from this parent, and small b from this parent. So do you notice here? Both the dose of capital A and capital B is present here. So the fly's eye color will be red here. So two white color parent is giving us the red color offspring, because from parent 1 we are getting capital B, and from parent B we are getting capital A, and both capital A and capital B is required for producing red eye color. That is why this offspring is red. The eye color of this offspring is red. So this is known as complementation test. So by the complementation of the two genes you are getting one character. Okay, so this is the example. If a characteristic is determined by only single gene, so this was case 1. This was case 1. Case 2, if a characteristic is determined by only one gene. Okay, so what will happen if there is a mutation? For example, these two are mutant parent. We only get mutant, that is white offspring. Okay, so as because this character is driven by only one gene, so in the offspring we are getting the white color. So by this we can understand that if a characteristics is determined by one gene or two genes. So from here what you can understand if two genes are present in different chromosomes, they will complement with each other. Okay, and if two genes are on the same chromosome, they will not complement with each other. So in order to complement, you need two genes on separate chromosome. So complementation needs two genes on different or separate chromosome. Then only the genes can complement and they can give us the wild type characteristics. But if two genes are on the same chromosome, they will not complement with each other. Okay, now here there's an example asking how many genes are present. So how many genes are present, how can you understand? Here plus sign denotes complementation, and you know complementation means the genes are on the different chromosome. Okay, and minus means no complementation and the genes are on the same chromosomes. So by that you can understand. So 1 and 2 there is a plus, so 1 and 2 are on the separate chromosome, but 1 and 3 they are on the same chromosome. 1 and 4 it is plus, that means complementation, so they are on the different chromosome. But here you can see 1 with 3 and 5, minus means no complementation, that means they are on the same chromosome. Okay, so 1, 3, 5. Then next with 2 we can see with 2 we can see 3, 4, 5 both are on the different chromosome. With 3 we can see 5 are on the same chromosome, because with 5 it is minus. So yes, we have previously also wrote that 3 and 5 on the same chromosome. Okay, for 4 we can say 4 and 5 on different chromosome, and 5 on different 5 prime of course on the same chromosome. So 1, 3, 5 on the same chromosome. At the same time 2 and 4 on the different chromosome. But point to be noted, does 2 and 4 on the same chromosome? We have to check that with 2 and 4. So we can see that with 2, 4 on the different chromosome. So 2 on different chromosome, 4 again one different chromosome. Okay, so number of gene we can see here 3. Gene 1 contain 1, 3, 5, gene 2 contain 2, and gene 4 and gene 3 contain 4. Okay, so by this type of examples or questions will come in your exam. Then you have to say that how many genes are present, like in a single chromosome, how many genes are present. So these are known as complementation group. So 1, 3, 5, these are known as complementation group. These 3 genes are present on the same chromosome, so they are known as complementation group, and 2 on different chromosome, 4 on different chromosomes. So as a whole there are 3 chromosomes, and 1, 3, 5 on the same complementation group, 2 on the different, 4 on different. So 1, 3 and 5, they are in the same complementation group. Now coming to the next example. So here again you have to do like that. So we have to see that what in the same chromosome. G with B minus, that means G and B on the same. G with, then F with A on the same chromosome. FA, FD, then E, C. So first of all you have to write about the genes which are on the same chromosome. That will be easy. So DNA, yes, DNA in the same chromosome we have previously wrote. Then B and yes, okay. So as a whole we can see that there are 3 chromosomes, okay. G and B are in the same complementation group, F, A and D in the same complementation group, and E, C in the same complementation group. So as a whole 3 complementation group. In group 1 2 chromosomes, in group 2 3 chromosomes, in group 3 2 chromosomes. Okay, now the next example is for you to do at home. You will do it, and if you face any kind of difficulties, you can always get back to me through my mail ID or through the comment section. I'll be very happy to give you a reply. Thank you very much, students, for listening. I hope you understood what is complementation. As I always say, keep learning and fight for your dreams. Your dreams will fight for you. Thank you.